Metabolic health is determined by a complex interaction between diet, exercise, and genetics, but what is often overlooked are environmental factors such as endocrine-disrupting compounds that can interfere with fat metabolism and even the programming of stem cells in the womb. Animal studies and epidemiological studies have been amassing good evidence for the influence of endocrine-disrupting compounds on the worldwide obesity crisis. It has come to light that a variety of plastics, herbicides, insecticides, polychlorinated biphenyls, and heavy metals can interfere with the hormonal and genetic regulation of fat cells. In experiments using mice, exposure to tributyltin in the womb caused irreversible changes to the developing mice. The result of these changes was mice that gained more weight eating the same amount of food. Tributyltin is a compound that was used extensively in the painting of ship hulls before it was banned. Tributyltin binds with receptors that regulate the expression of genes controlling stem cell differentiation and adipogenesis. Mice that were exposed during early stages of their development had more of their stem cells turn into adipocytes. This predisposes them to weight gain and obesity compared to mice that were not exposed. BPA, or bisphenol A, is a common environmental pollutant used to make plastics, and it is known to disrupt endocrine function. It has been found to have an influence on the same stem cell programming receptor as tributyltin. Once the fat cell is developed, BPA interferes with the expression of several important proteins and enzymes related to fat metabolism. On top of this, BPA has been found to impair the function of the thyroid gland and increase serum levels of obesity-related hormones such as leptin and ghrelin. Heavy metals such as lead, cadmium, and arsenic are known carcinogens and teratogens. A teratogen is a substance that interferes with the normal development of embryos. Pregnant mice that were exposed to heavy metals had offspring that had shorter gestational periods and lower birth weight. However, as the mice grow, they tend to have higher body mass and increased food intake as adults. Continued exposure to heavy metals in adulthood can lead to obesity through a number of mechanisms including epigenetic modifications, inflammation, oxidative stress, and insulin resistance. These three categories, tributyltin, PCBs, and heavy metals, are only a sampling of the wide range of substances that influence the programming of stem cells and disrupt endocrine function. Fat cells were once thought to be a simple storage site for energy, but decades of studies show that fat tissue is a complex organ that is highly sensitive to hormone signals and a site of hormone production. In this way, fat tissue is not just a storage site, but a sophisticated organ that is involved in the decision-making process of fat metabolism. While the effects that pollution causes on developing embryos is irreversible, it is extremely important to reduce our exposure to endocrine-disrupting compounds throughout adulthood. By eating organic foods, filtering water, and keeping up your overall health, you can protect yourself against the negative effects of endocrine disruptors. Unfortunately, these compounds are not easily broken down. It is because they are so persistent that we are still dealing with ambient levels of endocrine-disrupting compounds that were made illegal years or decades ago. Abandoned manufacturing facilities are a continuous source of these persistent chemicals that are now illegal to use. However, there are also compounds being released into the environment by modern industries that are simply unregulated. The regulation of a pollutant has to be backed up by thorough research, which can take years, leaving the regulators in a constant state of catch-up with industries that are causing human and environmental damage before substances can be regulated. Subscribe to hear more on polluted properties.